So far, we have discussed about cancer cell migrations in a brief in our previous videos, where we have understand what are the steps that are involved in the cancer cell migrations and how the cancer cell migration occurs and shift from one steps to another steps in brief. In this video, we will cover some of the steps in detail by comparing the functioning of the simple cell organisms and relate those functioning towards this cancer cell migration steps. Let us start with the figure on the left hand side. The figure on the left hand side uh, is the operation of these simple cell organisms, and this can be related to this cancer cell migration steps, where the chemoattractions helps in the cancer cell migrations. So far, in our previous videos, we have understand the TGF-beta helps the cancer cells towards acquire their EMT characteristics, that is epithelial to mesochemical characteristics, and that helps the cells towards migrations. However, we have not understood what are the changes that might have occurred within the cells. So in this case, we will consider an example of the simple cell organisms, how the cells migrate in the simple cell organisms. So the cell surface receptors, that is G coupled receptors, in this case are being activated, and the cells try to migrate from left to right directions. So when this G coupled receptors activated, it will trigger different signaling pathways such as P13K pathway and PIP3 pathways get activated, and some of the PIP3 independent pathways are also get activated. This will lead to the change in the cytoskeleton, or local activation of the cytoskeletons will cause, such as that is shown in this figure, and that will cause cell polarity, and that results in migration. So what happens in the cell polarity is that the cells that are trying to move, the portion of the cells that are on the front sides, that is the in the direction of the migration, and uh, they need to face the maximum resistance because of the cellular matrix present on in their front size. And in order to move, or in order to migrate on the right hand side, they need to somehow take care of the extracellular matrix. And in our previous videos, we have discussed how the cells release uh, exosomes that will help in the remodeling of the extracellular matrix, and such as degradation of the cellular matrix. And so these things are being released by this when these receptors get activated, and that will lead to this activation of the signaling pathways on the particular side of the cells, or that is in this case on the front side of the cells, in the direction of migrations, and that will cause release of the exosomes or other pathways that will finally lead to this remodeling of the cellular matrix and provide a way for the cells to migrate on the right hand side directions. And simultaneously it will also lead to change in the cytoskeletons, such as the cells will stretch in the right hand side directions, whereas on the back hand side of the cells, these P10 pathways get activated, and these P10 pathways will help in the compression of the cells from the left hand side. So if we can imagine both the things simultaneously, that is stretching of the front side of the cells on the right hand direction, and the compressing of the back side of the cells on the left hand side directions, both are on the left directions. So in this case, if both the things happen simultaneously, then cells will move towards right hand side directions. So this is how the chemoattractions plays a role towards migrations. Now let us quickly understand also this how this basement membrane invasion occurs. So if the cancer cells are developed in the epithelial cells, that they need to, in order to migrate those cancer cells, they need to pass, or they need to cross over the basement membrane, and as they are being surrounded by the basement membrane, and also blood vessels are also surrounded by the basement membrane, so they also need to cross the basement membrane near the blood vessels, and this steps is the intravasation steps, which is very critical for the cell migrations. So, and these things are uh, also been observed in the simple organisms, where the uterine cells interact with these vulval cells by intravasation of this basement membrane. So how this basement membrane intravasation occurs is that there is a factor, that is FOS1A, gets activated in the nucleus, that will lead to the upregulations of these genes, such as these affecting dynamics genes, cell ECM additions and ECM remodelings, and this will lead to this hundreds of dynamic invadopodia, and we have discussed about invadopodia in our previous videos. You can go through those videos to understand what this you can go through that video to understand what this invadopodia means, and you can also click the I link to understand about this invadopodia. And because of this invadopodia, uh, it will generate these recycling membranes, and the generation of this recycling membrane will cause this degradation of the basement membrane, and thereby this uterine cell walls can interact with these vulval cells after degradation of this basement membrane. and uh, and these things are just the zoom in view of this these simple organisms on this uterus to vulva interactions, and these things zoom in view of these things is in the part B of this figure. And what happens in the cancerous cells is that sometimes in the cancerous cells this natrin DCC signaling pathways get inhibited, and if it get inhibited, then the blocking of these hundreds of dynamic invadopedia will not occur, and cells can easily intravasate or uh, invade, intravasate or migrate through this basement membrane. So this is some of the basic steps how the cells can migrate. What are the changes that occurs within the cancer cells during these migration steps? We have covered two topics, that is either migrations because of chemoattractions, or how these cells can uh, how these cells can pass through this basement membrane. So if you need to understand more about the cell migration steps, each different cell migration steps in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.